I will be speaking on drug repurposing for the treatment of acute myeloid leukemia, current developments and challenges. So this is a kind of work which was my main theme of my current postdoctoral research in one of the lab here. So I'm not showing the current research as of now because of some confidential informations, but I will be showing some of the updates in the field and how the research is driving through in this field of drug repurposing for the acute myeloid leukemia treatment. So today's my presentation briefly have a few of the contents like I will be talking about how the global burden of acute myeloid leukemia and what are the key oncogenic signaling pathways for the targeting, and also I will be introducing to you drug designing and development process and what are the pros and cons of drug repurposing approach and current developments and challenges with drug repurposing for the treatment of acute myeloid leukemia, and also acknowledgements in the end. So we all know the tumors generally the before the tumor development normal cell divisions happen and the healthy cells will continue to the normally and the damaged cells will go undergo which cannot be repaired will undergo cell suicide like apoptosis pathways. But the cells will genetically modify and acquire the property of dividing continuously without any further stoppage in this division process, and it leads to a uncontrolled cell division leads to the development of tumors. So this could be a benign tumors or malignant depending on the how potent they are. Like and in the benign tumor generally they do not invade to the neighboring tissues, but they also can grow in size and cause health problems. And the malignant is the highly dangerous one, which multiply several fold before penetrating, and also it penetrates into different body tissues, leading to a serious health conditions. And
Dr.
the
Gola,
yeah,
sorry to disturb you in the middle of the session. Like your slides are not moving. Actually, we are in the first slide.
really,
Yeah,
oh, just a second.
yeah, yeah.
And now, no,
No, we're still in the first slide.
just a second again.
Yeah, yeah.
How about now? Now you can see the slides.
Yes, yes. Now it's visible properly. Yeah.
Okay, yeah. So as I mentioned, like the tumor burden is it's all over the globe, and different cancers are having leading to the global burden all over the countries here. So the majority of the tumors, like the respiratory things, like mostly lung cancers, and then colon cancers and stomach cancer, followed by breast cancer, and then leukemia has a impact of major impact over compared to other several organ cancers. But the leukemia has been a big challenge for the current community to get the proper treatments for the leukemia. So the another important factor that to be discussed is the drug resistance, and the treatment of cancer is becoming more and more difficult and requires new therapies because of the drug resistance mechanisms that are acquired throughout the therapy, as well as some of the mechanisms that are leading to the changes in the therapeutic drug pressure, as well as tumor heterogeneity factors and the tumor growth and tumor burden and the microenvironment around the tumor, like the several factors like immune system and tumor microenvironment also contributes to different drug resistance mechanisms that can make them very inefficient for the treatment of cancers. So before going to acute myeloid leukemia, so I will be briefly like to introduce you about the hematopoiesis process, where hematopoiesis is generally called as the process of formation of blood cells from hematopoietic stem cell and the bone marrow, and which is further differentiated into different lineages such as myeloid cells and lymphoid cell lineages, leading to the complete formation of different type of blood cells, including the lymphocytes as well as immune cells, which are important for our normal regular functions. So when you look at the normal blood as well as the leukemic patient blood, so the morphology looks like the most of the blood cells are occupied by immature blast cells, which are reducing the actual function of other normal blood cells. So this can be divided into leukemia, which is that cancer starts in the bone marrow of the blood types, and in case of lymphoma type, cancer that starts in the lymphatic system, and whereas multiple myeloma is a cancer that develops in the bone marrow and affects the plasma cells. So acute myeloid leukemia is one of the most common form of acute leukemias in adults, and the second most common form of acute leukemia in children. So there are more than 20,000 new cases every year in United States itself, and also it is mostly much more common in older patients, like older adults, which are aged about more than 60, 65. But there are also chances of getting this AML because of the prior history with myelodysplastic syndromes as well as myeloproliferative neoplasms also have been led to the development of AML in some of the cases. And the most common treatment for the acute myeloid treatment was from the last several decades. It was a standard 7+3 regimen consisting of 7 days of infusional therapy with cytarabine and 3 days of infusion with daunorubicin treatment. So the partially like the heterogeneity of AML is too much because it has several mutational factors that are developing to the disease. So the drug targets are very not very unique, and proper targets were still not available. And over the past 50 years, the standard regimen treatment did not change over several decades, as I mentioned. The 7+3 regimen continued till today. Like this is a major cytotoxic induction therapy treatment that was used, but over the last 10 to 15 years, the focus of research on the AML and then getting more knowledge on the pathophysiology of AML leads to the newer drug developments as well as getting approval of new therapy molecules for the AML treatment from the last few years. So there is a research that was talking about looking at the several patient samples, like around more than 9,000 different cancers, to understand what are the key pathways, key oncogenic signaling pathways that are deregulated in the cancers. And this is a snapshot of different pathways that are identified through TCGA genome data sets, and the mutational profile shows the key pathways that are belongs to receptor tyrosine kinases pathways, RAS pathways, as well as TGF beta, and then PI3K pathways, MYC pathway, and P53s, and cell cycle also plays very important role, and Wnt pathways. So if you look at the overall tumor profile and the role of different signaling pathways in the development of those cancers, you can see the AML has the majority of cases belongs to the receptor tyrosine kinases as well as RAS kinase pathways, and the cell cycle also got majorly affected with the AML features compared to other signaling pathways. But still, there are molecules and drugs that are still targeting the other pathways, that has PI3K and Notch pathway and Wnt pathways are still playing very important pathways for drug targeting in the AML. So the other emerging druggable targets for AML treatment, the recent evidences also shows that in addition to those signaling molecules, the mitochondrial signaling also like plays a very important role, can be a druggable target for the treatment of acute myeloid leukemia from the recent evidences. So some of those molecules are IDH2, like associated dehydrogenase from the mitochondria, also have been tested and then found to be very effective for the AML treatment. So some of the drugs are still under investigation, and some drugs are already approved for the AML treatment. So in addition to the mitochondrial pathway, there is another key factor that is AML resistance, drug resistance is leukemia stem cells, which are very less fraction of the cells that are converted into leukemic stem cell properties in the bone marrow. And after the initial induction therapies, so the remaining leukemic converted stem cells again may have a chances of getting relapse, like the disease relapse second time is very severe to treat, and it is a highly drug resistant relapse actually. So The recent therapies also focusing on targeting this leukemia stem cells, especially for getting those relapse treatments. And some of the treatments are majorly focusing on using the cell surface markers such as CD123 and CD47 and CD33 markers for targeting those leukemia stem cells, especially without disturbing the normal hematopoietic stem cells, which are essential for our normal blood formations. And one of the agent that I should talk here is venetoclax, which is 2 years ago got approved for the modifying the apoptotic pathway in the mitochondrial in the leukemic stem cells especially. So this is the overall snapshot of current drug target drugs which are under investigation, and some of them are already approved, which are with major tick marks. So these are the major pathways that are currently used for targeting treating to the AML patients. So mostly the apoptotic pathways, like depending on the BCL2 and MCL1 and hedgehog pathways, and as I mentioned, receptor tyrosine are the most promising druggable targets from the TCGA data sets. And there are also some mitochondrial one, as I mentioned. These are new emerging drugs that are approved, and some of them are still under investigation for the this one. And also, as I mentioned, immunotherapies for targeting, especially the cancer leukemic stem cells. So that is also under very seriously under investigation for different levels, very the clinical trials. So as I mentioned, the drug development and designing process involves very several multi-step process involves from the target identification, and then from there it will go to the hit identification, lead optimization, and then before going to the human trial, the efficacy studies will be performed in preclinical models, and clinical trials performed in different stages to confirm its safety as well as efficacy in different group of patients and populations across the globe, and then the final approval comes into the market. So this overall process of identifying a drug, even from simple fever like a paracetamol to the very complicated disease such as cancer treatments. So everyone must go through this entire pipeline involving from the beginning, like target identification to the phase 4 post-marketing surveillance approval. This is the final pipeline where it has to go through several years, and a lot of money is involved in this drug designing process. Like overall 40 to 50 million for one drug molecules to enter into the market involves a lot of efforts and a lot of years of hard work. So when it comes to the drug discovery process, so generally we use target-based drug discoveries. We will, as I mentioned, people look at the one gene and then think that this might be important target for cancer treatment. Like, so then they use that some molecule that to inhibit those particular pathway, and then look for the endpoints that this could be a very helpful molecule. But this is all just a pathway that we really don't know how the cells behave in the in vivo, like. So the real picture looks like something very complicated. So we might think that one molecule can really inhibit some of the cancer pathways, but the real picture is that one molecule that we are going to target may have several interactions inside the cell and may lead to different off-target effects, and also different adverse effect leads to its withdrawal from market. This has happened with several times with several molecules. So the phenotypic observations generally very helpful, such as like 30% of 400 compounds profile show new beneficial biology, and also up to 90% of the new indications are driven by on-target activities. So as I mentioned, the biology is very complex and there is a tremendous amount that is not understood. And the phenotypic screenings provides an opportunity to identify new clinically relevant uses of existing molecules driven by an action of unknown molecular target. So that's where the drug repurposing, also called as repositioning, enters. So drug repositioning and repurposing is generally is the use of an active ingredient or drug in an indication other than the one which is initially marketed or approved. So this actually has gained very attention from last 2 decades. Though some of the first few examples of successful drug repurposing came through the like as we know, paracetamol, like thalidomide, sildenafil, and dimethyl fumarate. So this drug repositioning relies on two scientific bases. One is the discovery through human genome elucidation, such as some diseases share common biological targets. Like we can expect if a drug is working in one of the disease where pathway and the targets are known, we may expect that this drug may act in some other relevant cancers where that particular target is really involved in the pathogenesis. And the second approach is where concept of pleiotropic, having multiple drug effects. As I mentioned, what we thought like it might be targeting one, but when it comes to the real scenario, it might have multiple targets in the cell that may lead to its another multiple effect properties. So this pleiotropic, the pleiotropic property of these drugs are very helpful for identifying a new function for the existing drug that helpful for the shortening the drug discovery process. So if you look at some of the examples of these repurposed drugs, so one is the, as we all know, aspirin, which is coming the 1899 as an analgesic, but later it got 1980s, it got antiplatelet property, as well as now this is under testing for anti-cancer drug properties. And another one is thalidomide, which was banned in 1962 for its um, adverse effects, and then later it was approved as a drug for the leprosy treatment, and then later in 2006 as a treatment for multiple myeloma. And the other standard of drug that is useful for is sildenafil, which is entered in the market as a antihypertensive drug initially, but later it got withdrawn from the market because of its extra additional effects. But later the adverse effect got made it as a main function and later entered into the market as a 1988 for a erectile dysfunction treatment. So if you look at those repurposing treatments and the drugs which are successful in the repurposing, that is has encouraged so many researchers to focus on the drug repurposing to shorten the drug discovery process and identify a new drug for the diseases which are not curable and finding in new treatments. So the advantage with the drug repurposing is that it may not enter into the first phase of the drug discovery pipeline. So it really shortens the path because as we already have the safety profile of the drugs which are already approved in the market and which are already approved by FDA for the treatment of some of the disease. So we already have the safety and efficacy profile for the drug. So generally what are the new uh, indication where we want to test the drug, it has to be tested, comes to the phase 1 trial, like start with the only clinical development process where it has initially tested in small population where just to check its safety, and then it will tested in some of the small patient samples, patients where we can be tested for efficacy, and then later it will tested in the phase 3 where large number of patients are involved to just test the both safety and efficacy in a different group of patients, and then finally once it get approved, it will enter into the market and phase 4 surveillance will be observed. So this kind of drug repurposing is now getting more attention because of shorter drug approvals. So some of the cancer drugs that are repurposed, like non-cancer drugs that are repurposed for cancer therapy, are listed and reviewed by several papers. So here are some of the drugs which are non-cancerous, but they are repurposed and tested effective against for different cancers under investigation for the treatment. So those includes like antihypertensive drugs and then beta blockers, anti-diabetics, and also antimalarial drugs and antibacterial drugs. So these are like several kind of molecules which have been found to be effective in the preliminary studies in vitro, as well as some of the in vivo studies are very encouraging for their testing in the in vivo models and also in the clinical trials. So one of the drug that was today I will be talking on the antileukemic drug, that is niclosamide, which was recently found to be effective for the AML treatment. So this is the paper in 2018 where it showed this is a antileukemic drug which was approved several years ago for the treatment of in the occasions where we have parasites disease. But here it has been found to be new use where it has treated found to be effective in both in vitro as well as human xenograft models. It was found to be very effective in controlling the AML disease, and it was found to, from the genetic mutational screening, it was found to affect mainly the PrEP pathways where it has a activation regulation through receptor kinases lead to the final modulation to the transcriptional factor and then leads to the gene expression changes that are very essential for modulating the key cell signaling process. So the other drugs that are repurposed for AML treatment are reviewed and under clinical outcomes are outlined here. So one of the drugs that are will be hydrochloroquine, which has gained very recent attention for the SARS-CoV-2 treatment, but unfortunately it has gone failed for the trials. But these kind of drugs which are still under investigation for AML treatment, and then atorvastatins, these are anti-hypersensitive drugs which are also have been tested for different cancers, including AML, have been found effective. And the other drug that I was currently working on, metformin, in our lab. So the findings are very preliminary but very encouraging in our lab for the AML treatment. And also we have found metformin was found effective with other standard of care therapies such as citarabine and venetoclax. So those results are very encouraging. So we are still pursuing those results in different AML models to confirm its efficacy, and also using primary patient samples in the uh, check the efficacy in those ones. And the HDAC inhibitor like valproic acid also was found to be very effective. And there is a new class of molecules like even plant-based directed digoxin also was found to be under investigation for AML treatment. So this is a table where it is summarizing the clinical outcomes from the recent clinical trials. So some of the molecules which are repurposed have been found to be not very effective in the initial phase, the clinical phase trials. And one of the molecule that thyroid was found to be very toxicity, and some of the molecules have been found to be very slow in the disease treatment. But these are found to be very in single agents, but the combination could be very helpful of these agents and can be tested further in combination with other standard of care therapies in the low doses for decreasing those side effects also. So the ongoing trials are still under investigation for other class of molecules are still under investigation for the AML treatment. So that's overall my summarizing of those drug repurposing advantages and then current developments. So, and finally, I would like to acknowledge my current mentors like Professor David Claxton and R.T. Sharma for providing the key reagents and resources for my research on the AML, and then my institution like Penn State Cancer Institute, and then funding from National Institute of Health and Four Diamonds supported from PDX and Therapeutic Score is highly acknowledged for their internal grants as well, funds for the research. Thank you all for your time and then patience. Please let me know if you have any questions.
Upendra, this is Professor Ramarao.
Hi.
Hi. It's very nice topic, and repurposing. It's